Hey everyone. So in the previous video we have discussed about superoxide dismutase and antioxidants which disproportionate the catalyzes the disproportionation of superoxide ion. So catalase and peroxidase are more or less same. Okay, there is a very little difference between both the things. Catalase is an antioxidant that breaks hydrogen peroxide H2O2 into oxygen and water. We know that there is a process in our body, the process of converting oxygen to water. So this process has so many intermediate steps due to the free radicals generation. There is a production of free radicals and the reactive oxygen species. So H2O2 is hydrogen peroxide. H2O2 is not a free radical, but still it is highly reactive because it is considered as a reactive oxygen species. Okay, though it has not any kind of free radical, but still it's very reactive. So to destroy this, to catalyze this H2O2, we have present catalase. Okay, it is an antioxidant which catalyzes the disproportionation of this H2O2. So let's find out what are the things. Firstly, we have to know the structure and the mechanism, how it's working, how the equations are working in catalase. So basically we are going to study about the beef liver catalase. Why we are studying this? Because only this much information is available in the Wikipedia or this much research has been done by the scientists. That's why we are only studying about beef liver catalase. Okay, so catalase is a tetramer of peroxidase enzyme. It is found in all the living organisms. It decomposes the hydrogen peroxide. One catalase converts millions of molecules of H2O2 to water and oxygen in each second. So here you can see the power of this catalase. In each second, one molecule of peroxide can get converted to water and oxygen, right? So if you notice the structure, of this, here in the resting condition high spin Fe3 resides here. This is very common to you if you all know the structure of heme protein, hemoglobin. This is 4-imidazole nitrogen. Okay, in the axial position we have H2O molecule, and in the bottom we have the phenolate group. Okay, so this is in catalase, one axial position is occupied by the water molecule which replaced by the H2O2 in the catalytic reaction. Now let's study about the mechanism. So I've jotted down each and every steps in a theoretical form so that you won't face any kind of problem. So catalase catalyzes the disproportionation of H2O2. Catalase follows a two-step mechanism. There is two-step mechanism to decomposes hydrogen peroxide into water and oxygen using a heme based active site containing an iron center. So step 1, formation of compound 1. Now what is this compound 1? We will see here. Resting state of catalase has Fe3, which is known as ferric, bound to the porphyrin ligand. Okay, hydrogen I am considering you know what is porphyrin ring because you have completed the chapter hemoglobin and myoglobin. I am considering you know all the things because these terms are very important. And hydrogen peroxide binds to the Fe3 site. Okay, the interaction leads to the oxidation of Fe3. So here is this, in the examination you're going to write this as a first step. Okay, so let me make you understand what is the saying. This is Fe3, okay, plus H2O2. The interaction leads to the oxidation of Fe3 to Fe4 is equal to, here P plus represents the porphyrin ligand undergoing oxidation. Okay, this is porphyrin in the first step. This is the reactant side. We do not have any kind of oxidation here. In the second step, this iron is undergoing oxidation. So this will Fe3 will get converted to Fe4, and this will have a plus sign over here, and O is oxoferryle forming. Okay, it's compound 1 and H2O is releasing in this case. Okay, I hope this part is clear to you. Here this compound 1 is this. Here P plus represents the porphyrin ligand undergoing oxidation. Compound 1 is the most active intermediate in the catalytic reaction. Means the process won't stop over here. We're gonna have the next step. Okay, what is the next step? Step 2, the reduction of compound 1 and oxygen redrawn. All right, redrawn, not it is oxygen releases. ठीक है. So another molecule of hydrogen peroxide, now another molecule of hydrogen peroxide will react with compound 1, reducing the porphyrin radical and forming compound 2. Compound 2 has iron 4 with an oxo ligand but no porphyrin radical. So here is a compound 1. It is again reacting with another electron of you can say here it is H2O2, because it is another molecule of hydrogen peroxide, which is represented by this E. This is again compound 2 has iron 4 with an oxo ligand, but in this case now we have no porphyrin radical. P plus नहीं है इसमें, ठीक है. So this is your compound 2. Okay, write this as a compound 2. So here in the first step we get the compound 1. In the second step we will take compound 1 in the reactant side, okay, and H2O2. Then this is again going oxidation and we are getting Fe4. All right, now you will say we already have Fe4, so in this case Fe, this is iron is not going any oxidation, but here reduction of compound 1 is happening. So compound 2 has iron with 4, which is remains same with an oxo ligand but no porphyrin radical, as this porphyrin radical undergoes the reduction process. Okay, you have to learn all these things. Step 1 is this formation of compound 1, how it is formed, like oxidation of iron and porphyrin ring and porphyrin, and this step 2 reduction of compound 1 and oxygen gets released out. Okay, in step 3 we have this Fe4PO. This is your compound 2. You can name this as a compound 2 plus this gives Fe3P. Now compound 2 is here reduced by an electron returning to the iron center to iron 3 Fe3 plus. So here this E minus is representing that this compound is getting reduced. So this Fe4 iron will become Fe3P. Okay, this regenerate the enzyme in its resting state, ready to start another cycle. Now when it comes again to Fe3P, the cycle will goes on continuously, because this is the first step here. Then again H2O2 will come and this will break down like this. So basically there are only 2 steps happening over here. First is oxidation, then is reduction, and this can be say this is intermediate step. The last step, this the reduction of compound 2, which is ultimately we are getting the same reactant as we get in the where we have started. Okay, so the cycle allows catalase to efficiently decompose H2O2, that is hydrogen peroxide, into water and oxygen, protecting cells from oxidative damage. I hope this is clear. If there is any kind of doubt, you can ask me. Please have a screenshot of my notes. Thank you.